All right. Good afternoon, everyone. Thanks for being here. It's my great pleasure to introduce Dr. Oyetola Oyebanji, who is a postdoctoral researcher working with me and my PhD student here, Miyata Andrea Manohara, as part of our NSF grant entitled NSF DEB NERC, Subtribal Classification and Generic Delimitation among Eastern Hemisphere Ironweeds, Vernoni compositae. So just to give you a little bit of background, Tola started his postdoc at the end of March this year, moved here to Fort Worth with his family, and so you'll see him around. He's mostly in his office lately in the Barclay suites, doing a lot of computer analyses on data that we have already collected, but you'll see him around in the team room or in the lab and elsewhere. Tola, just to give you some background, he received his Bachelor of Science in Forestry Management from the Federal University of Agriculture in Nigeria in 2010. He completed his Master's of Science degree in Botany at the University of Lagos, Nigeria in 2015. He completed his PhD in Plant Molecular Biology and Biotechnology from the Kunming Institute of Botany in 2021. In addition to his education, he also has served as a herbarium curator at the University of Lagos Herbarium from 2011 to 2017. He was also a postdoctoral associate at the Kunming Institute of Botany from 2021 to 2023, and a postdoc at the University of Arkansas most recently between 2023 and 2025 before starting here at Britt. So without further ado, I will pass this over to Tola, as we affectionately call him. And also just want to mention, after the talk this afternoon, we're planning to have a little welcome celebration because we haven't been able to do that for Tola yet at Rodeo Goat at 4:30, between 4:30 and 7:30 this evening. So feel free to come on by. And thank you.
Yep. Thank you, Tomagan, for that introduction. I'm really excited, and I really appreciate everyone here in person and online to attend this talk. I'm kind of like excited to be here because my first visit here, I was like, being blown away by the kind of facility that, we have here. And, I really look forward to work with everyone. And today I'll be talking about some of the things that actually made me more curious while I was starting my career. I'm always curious about why do we have one species in this place and then we couldn't have them in this region. Looking at those primitive information kind of like led me to a lot of things. And in addition to what Tomogana just said, I just want to quickly give us a kind of a snapshot of how has it been with me. So I've been to 3 different continents, and I've explored, gathering knowledge from different angles, both in analysis and field work, and of course, using different types of model organisms in my career. And also, one of the most fascinating things about my work is that I really enjoy going to the field. And just like 2 weeks ago, I was just having a discussion with Anna while we were walking through the library, and I ran into flora of West Tropical Africa. And I was like, oh, this man here is late right now, actually was part of those who assembled the flora of West Tropical Africa. So learning from him is actually kind of a great blessing for me in my career. And still in that same vein, I was able to go to the field with students and, explore different part of the work here. I always like this picture because you can actually differentiate me from my student here. So, so I always really like that picture. And like he has said, I've also done a lot of little, creative activities over the years. And these are some of the things that I put across for, exhibition and also for teaching. And so over those times, I just want to talk about little things that I've done so far. Like, taxonomy is very important because bad taxonomy can kill. And I'm actually putting forward micromorphology for us here to see, not because they are actually telling us that we could actually use this information to delimit species. If you agree with me, ain't this look so beautiful? Architecturally, they really look so aesthetic. And then I also look into some conservation approach at different scales. So I'm just using this as an example of, predicting suitable climatic for tree species, economic species, endangered species, and of course, what are the factors that actually affect species composition? That has been the bedrock of my, research. Like I always want to know in this particular ecosystem, what has been the main factor affecting the species distribution. And my recent work on tumbleweeds, which is invasion genomics, also tells me that over time, genetic components of species, individual species tend to change through time. And of course, this could actually help their aggressiveness and how we could control them in the recent world. Shifting to breed, I would be working with the ironweeds from the Eastern Hemisphere. And I'm really excited taking up this journey with Morgan, Mihatha, and folks from Q. We have series of meetings on this. And of course, I really look so excited about what we're gonna get from here. And moving forward, just to tell us very little thing of what we are expecting here. So this is the focus on the Eastern Hemisphere. And the data source is gonna be from the herbarium and the field. And of course, let's think about anything called integrative taxonomy coming from the morphology, both the macro and micro, and as well as the genetic aspect of it, and the phylogenomics aspect of it, capacity building, that is to train students or interns on some analysis in this group, and of course, disseminating our information via papers and workshops. And so let's think about anything that has to do with plant science here. We talk about basically we are looking out for seeing if we can have new species or new lineages being ascribed within this group. And then we talk about reticulations, trait, and biogeography of this group. Being summed together, my interest is I could categorize it in 4 different groups. So from ecology to taxonomy, so microevolution is when we think about evolution within a very short time, and of course, within a very small species. While macro is actually on, involves more species and of course across a very huge timeline. So let's talk about this. On this side, we have the epoch periods here. And on the left side, we have the evolution of, organisms. And we see here, this moves from past to present. And what we need to pay more attention to here are 2 basic events here, which happens to be extinction period. And this actually reshaped the evolution of plants, or let me say biodiversity across the globe. We will talk more about it later. Coming from the ecological aspect of it, you could see here that this is the current continental plate that we have right now. And in that sense, we could see the evolution of, mix of forest, non-forest, and the forested regions. Then next to it is the global temperature surface. And this shows like at some point we have humid temperature, and also climate conditions and dry climatic conditions. All of these are functions of the oscillating, or let me say the temperature trend. Importantly here, we could see that this uphill of temperature here actually coincided with the evolution of the angiosperm, when angiosperm began to radiate sporadically globally. And that was also supported by the crown age of, of angiosperms, how it become, very common to what we have right now. Within a very short period of time, I use short period of time because in the previous slides, I talked about like 200 million years ago. Now I kind of compressed this to 60 million years ago, just for us to see how geological or paleoecological events take place. And we will start with this from, from temperature. We can see, just as we have it in the previous slides, where we have the temperature just declining down to the present. And here, when the temperature began to decline, then we have the Antarctic ice sheet forming here. Another thing to take note here is that we have different geological events here, and one of it is the India-Asian collision. And of course, let's talk about the Isthmus of Panama. One thing I want us to carry with us here is that these events could actually serve as a migration corridor, or they serve as a barrier for species dispersal. So these are going to be some of the reasons that we are going to really talk about, as we talk. Now, we both agree with me now that we have heterogeneous species distribution all over the world. And then here, I kind of like model in a way that we have like six regions here and migration through time. What happens? We see that some species were able to move from this region from the past to present. Fortunate for region A, it had one species towards the present. And of course, we see a case of C and D, they could not get through. So what happened at that point in time? There is what we call dispersal. And that could be as a function of niche equivalency, high adaptation rate for that species, or high dispersal rate for them to move from this region to this region. And what happened to this other one, which is extirpation, that is extinction. That means probably, they could not disperse. Bad ecological conditions and the likes must have actually made them to lose that. And then I come to the conclusion that each biogeographic region plays significant roles in species dispersal. Either they serve as dispersal source, or they serve as dispersal sink, as we can see here. Putting all of these things together in terms of community, then we have phylogenetic diversity. Plus or minus, as a biological organism, what plays a role in our population is rate of birth and death. And that comes into speciation and extinction rate here. So I imagine that one of these lineages actually got extinct. So what happens next? Then we see that there might probably going to be low diversification rates in this lineage compared to this part. And all of these things that I put together is just called natural theory of biodiversity, because all of these things are stochastic. They just occur randomly. And in plant biology, or let me say in biology generally, there are different hypotheses or concepts that has been used to do this. As you will agree with me, the very first one is there any link with all of this plant biodiversity with Earth history? Another one I'm going to talk about is out of the tropics hypothesis. That is, species that originate in the tropics, they kind of have the opportunity to radiate in the tropics, and then they go into the temperate. The third one I'm going to talk about is the cradle and museum. This is quite very interesting because a lot of literatures have actually argued a lot of things about this particular concept. And to cut it, just to go straight to the point, cradle means region of speciation. That is where the species tend to have new beings, or let me say new species. And of course, museums, as we all know, museums that harbors old things, that is old lineages there. So the last one I want to talk about is the very popular approach, which is classic biogeographic pattern. Like, how do these species actually move from one place to the other? For us to be able to understand this, actually, we need to have a well densely sampled phylogeny that will be dated for us to be able to understand the movement of species in time and space. And with that, I'm able to introduce to you legume as one of the model that can, be used to test some of these hypotheses. Legume, we can see this huge number of species that it has. Of course, it's one of the most successful angiosperm lineages. And of course, they are, morphologically blessed with different types of floral architecture and the likes. And I won't bore us much about the taxonomy here, but I just want you to know that it's still undergoing revision, because now, before we used to have 3 subfamilies, now it's 7. And the clade I'm working with here is in the subfamily Papilionoideae. And for the purpose of this presentation, I'll just refer to this Millettioid Phaseoloid Clade as MP clade. And then it also has 6 lineages. Surprisingly, these 2 lineages actually have become the most notorious in that group. However, I won't really say much about that in this presentation because I just want to, cut it down to the most important part. And another question I often answer is, why do you actually pick this clade? And I tell them, this clade includes several economic species that we consume, let me say, like every other day. So it's worth knowing what is really going on with their distribution, how we have them in the current state where they are. Here you can see I mapped the distribution of the species here. And of course, the darker the color, the higher species distribution that we have in there. And we could see like, oh yeah, we tend to have some legume food in this part of region compared to the northern part. And individually or as a unit, I kind of like try to also show the distribution pattern. And what we can see here is that some species or some lineages, they are more in the southern hemisphere compared to this that has actually peaks both in the northern and southern hemisphere. So the way they are actually responding to different ecological conditions in their evolutionary history. So as at the time I was doing this work, there was really no phylogenetic well-resolved tree that is directly sampled in this group. And of course, because that is not available, we were unable to understand how these species have been able to move from one point to the other. And it comes up with 3 different objectives, which is, of course, the primary one is to, reconstruct the phylogenetic relationships within this group, and using that as a plugin to understand the divergence time, and moving forward to see how the species have actually moved from different places to what we have right now. This is just like a compressed methods for everything, and I'm just going to go through it. Mostly the kind of marker that we used here is generated from the chloroplast and nuclear ITS, and these are the major analyses that I performed. And just for the DNA samples, it went through different types of downstream analysis, like assembly and the likes. And here, the tree has like less than 700, more than 700 species in it. So it's kind of like huge. So this is an ML tree that we just reconstructed for this group. And one of the things that I want to point out here is, number one, we now have about 6 lineages in this group as compared to the conventional 3 lineages that we have, which means that the more you increase your sampling size, the more you tend to have more stories to tell in the group that you work with. And also, these 2 tribes actually nested in a very strongly supported clade here. And it has been an argument that instead of giving this tribe as a tribe, we could actually zoom them into like a subtribe of this particular lineage in order to kind of like enhance this group. However, I wouldn't really say much about this, but this is just where we are at this point. And it's most obvious that now we have a more visible, lineage classification within this group. And of course, we found 4 monophyletic tribes, as we can see on the compressed tree here. And this is also the tribes I was trying to mention earlier on, that probably we need to just put them in one, as you can see them on this lineage here. And then, of course, it's just going to be phaseoloids. And then what are the types of retribution events actually driving this group? So this is kind of the forward-thinking approach that probably if I have funding for, I could actually, put it forward. Having this now gives us the opportunity to now have how old is this group that we are talking about. And to do that, we need to plug in that same tree I just showed right now, the ML tree I just showed right now, and into a package called, 3PL. So using this algorithm, we need to calibrate the stem. And this is how it's been done. We just plug in the tree into this algorithm and then set these node edges. So these node edges were actually from 2 different sources. We have primary, which is from fossil, and secondary, which I extracted from different papers. So doing all of these things, plugging it in here, I was able to estimate the age and as well as the diversification rate. Down here is just a typical example of a fossil, how it looks like, and how you could identify them for your work. There are several databases that you could actually make reference to get all of this information. And then, so this is the dated tree. And the very first thing I want to talk about here is that the age of this tree, of this group I'm working with, this MP clade, actually originated around 61 million years ago during the Cenozoic. And if you remember, in the very second slide that I showed, I talked about the mass extinction, the first and the second extinction rate. And then we see here that there was mass extinction around that Cretaceous period. And of course, that led to the extinction of all those large mammals. And now we have legume evolving and really gallivanting everywhere. And then from the climatic aspect of it, it means that at the early part of this group, it coincided with when there is high temperature, and then towards the recent, it began to, to decline towards the recent. So which means that there is a kind of like a slow integration between, or let me say diversification from the very high temperature to lower temperature, which would actually mean that this group may actually have different evolutionary rates for them to be able to adapt to those changing climatic conditions. And I think we can remember from from some of the previous slides where I showed the non-forest region, also the dry and wet humid climate. So all of these things kind of play in their diversification. So this is the diversification pattern that I modeled from the model from BAM. The warmer the color, the higher the diversification rates. And what I want us to see here is that we could see that right when this temperature began to drop, we have very high diversification rates. As I was saying, that when the temperature began to cool, these species just try as much as possible to change in their evolution. And of course, I'm not surprised with that because there are several works that have actually tell us very few things about why these species can survive different ecosystems. And here is the temperature as I've been showing in previous slides, and we can see here. Another evidence from this work is that all of these round dots on the phylogeny usually shows that there are several evolutionary events going on, perhaps adaptation to ecology, perhaps leading to new lineages, as we can see here. This particular lineage is just a whole bunch of tribe. So probably the evolution just led to this, and within some of these groups, we have periodic adaptation between them. And as I've said earlier on, previous work have actually detected several whole genome duplication. And what this means is that gene duplication is one of the most evolutionary events that happens in biological organisms. And in this case, it actually have the possibility to link with the evolution of nitrogen fixation gene in legume. And with that, even when they find themselves in a very harsh condition, they were able to still survive. And that it's a big plus for them to have very high species richness and also being able to colonize different habitats. This is showing how the birth and death plays between this particular group. And of course, when the extinction rate is very low, we expect that the species richness is going to be high because we have less extirpation within the group. And that is what we are seeing here. And below here, it's not surprising to me as well, saying that there was no stoppage, no delay at all in the lineage accumulation of this group because they were able to adapt easily through time. And the point I'm bringing out here is that when there was a a sharp decline in temperature, then we had a spike in diversification rate here, which means that this species, as I've said earlier on, they kind of like struggle to be able to adapt to this changing environment towards the recent, and that is why we have them, being able to multiply at, a very high rate. If we think about this in the long run, phylogenetic is not just for us to study taxonomy. It also tells us a kind of approach for us to be able to understand our world, irrespective of the organisms that you're working with. Secondly, it also talks about the species distribution patterns, how we could relate geological events, what has happened in the past, paleoecological events, paleoclimatic events, and how those has interplayed and actually formed their current distribution that we have right now. And back to my first opening question, this actually answered one of my curiosities, that some of the species that have restricted or narrow distribution may actually have very low adaptation rates compared to those that are generalist, that have very broad adaptation rates and also can move from different place or the other. Now we look at it from this aspect. The next is to look at where did we actually have the species originated from. It's also an hypothetical approach to see how we move from point A to point B without, let me say, like, there must be a vector, right? Either by wind, either by car, or whatever it is. So in this case, I'm always very curious, because when I look at when I first studied the distribution of this species, I always wonder, like, how can this just happen? Like having same species in different places. And so to do this, I kind of like look into different models as well. And like I said, this is, I mean, a lot in this, but I just kind of make it a little bit more, simplified for us to understand. And while doing that, I kind of like try to create a model based on closeness. Regions that are close to each other, definitely there is a very high dispersal rate to be compared to those that are actually far apart. And to do this, I divided the continental plates into 6. So in this case, I did not separate Madagascar. So I lumped Madagascar into, into Africa. So sorry, Beata. So with that, I was able to just, based on time, and actually plug this in basically based on the geological events that happened at those different time frames. And then I used the dispersal extinction cladogenesis method to do that. So there is an algorithm that's going to tell you, actually, this is the best method for you to use. So we didn't just decide on what to use. And yeah, this actually gave me a nightmare to make it look this way. I mean, I spent like weeks on it because, this is more than 700 species. And of course, I'm trying to see how can I actually communicate stuff from this. So I'm going to try my best just to make us understand. From the base here, actually, we know it's just a time frame, right, that we have before. And this red or pinkish color here just talked about the Bering Land Bridge. What that means is that at some point from 61 million years ago, there were periods where we have connections between different islands, different continents, or whatever it is, either on a very large scale or at a very small scale. And this is one of the most common one here. It's called North Atlantic Land Bridge. And that is very specific about between Southeast Asia and North America. And here I have boreotropical flora. This is when we have species from the northern hemisphere that were moving down to the southern hemisphere because of the ash climatic because, it's getting freezing there. So they have to move down. And especially this barotropical hypothesis is one of the hypotheses that's being used to describe species that originated just, like maybe in recent time that doesn't coincide with, any significant tectonic collision or events. So these color codes actually, they correspond with the designated biogeographic areas, as I've said earlier on, which actually ends in Australia. But this was actually part of the result from the modeling. And what we want to see here is that we could see that this species has actually occupied different continents. And we could see at this point, this is the origin of this species, 61 million years ago. And hypothetically, it originated in Africa. And this other part of Africa means that several lineages of this group also originated in Africa. As we can see, the majority of them having this, can I say brown or whatever it is, colored here. And on each of these nodes shows the age of these lineages, right? So it means that we have the origin of this species from Africa and then moved down to Asia, which also led to the origin of two lineages of this group. And the summary of what I would say here is that this is a tropical species, and it has actually maintained also, the cradle as being the tropic and also the museum as the tropic. That is, old lineages are in the tropics, and also the new lineages, I mean, the recent one, they are also found in the tropics. The next we want to look at is how did this lineage actually moved. As I've said earlier on, so this box here is showing the origin, which is, Africa. And this is, let me say, this is the most recent continental plate, as I've said earlier on. And here I just kind of model how do these species tend to move. And we could see here that some lineages, like 2 and 4, they were unable to do what? To move outside Africa. That means they have, and they are endemic to Africa. In this particular group, they could not actually overcome also barriers for them to be able to move to other places. So there are other factors that we will want to look into to explain this situation. And then let's look at 5 and 6. They are just all over. So from here, we could talk about them being able to leverage the classic biogeographic approaches available, which is dispersal mostly, and also vicariance, for them to be able to, to move to this far. And then I also love this image a lot because while they ask me questions, and I always say, yes, they disperse. And then my boss asked me, so how? And I was like, okay, human can also do the same. And then it was like, okay, just put birds and monkeys there. So, and generally, Legume is known to be, I mean, I'm sure you know, is very cosmopolitan. And these are some of the, background processes that make it to be that cosmopolitan in what we have in the present world. Starting with these arrows, it shows the number of dispersal events. The thicker the arrow, the higher the dispersal events. Even though Africa is actually the main hub, right, which is the origin, but Asia seems to, give more even to Africa, which means it looks like, a two-way, there's always a two-way, a reversal dispersal approach in this event, in this group. And as I mentioned earlier on, this is a typical example. I was saying something about Beringia the other time. So Beringia is just like the land bridge, that was actually available during this period that allows biotic exchange from North America to Asia and also from Southeast Asia to North America. And also this pathway has also functioned as, migration corridor for even large mammals, for them to be able to, move between those continents. And one of the stochastic approach that legume usually use to disperse is actually long distance dispersal. We can see here that there is a dispersal from South America to Asia here. It didn't pass through, some passed through south to north, and then to Asia, Southeast Asia. While we see some that move directly from Asia to South America and also, vice versa, which means that some species, because of probably their fruit morphology and also the presence of birds as a dispersal means, of course, we all know legume, just a little hard, they're going to sprout. So they were able to sail through, overcome these barriers, and then they move directly to these places. So this graph actually was modeled according to the result, which means that some species could actually go to get to the new ecosystem or to other places, either by going through the closest places. Example is what we have here, like, it goes through from South to North America and then, come here. And of course, it's going to be like a circle from also Asia down to, Australia. Some made it, and we can see here that only Europe could not give out samples here. And of course, I would say probably the sampling is still a little bit biased. Probably if you have more samples from Europe, this might actually change the story. And of course, we could not actually overlook the contribution of these guys in the process because, legume, everybody consumes legume. And the way these guys covers the global space is really unimaginable. And that's why I kind of like come to conclusion that, of course, I mean, you can see they were being caught red-handed here, eating some of these stuffs, and then which also help in the dispersal of this group. And here I want to talk about, like I said, the classic biogeographic pattern that I talked about the other time. So without dispersal, this species would not actually be able to achieve the current present day distribution. I imagine if the seed happened, of course, many of us know legumes, there are different types of beans here. Imagine it having like maybe in big and also have delayed germination rate. So which means that probably dispersal may not be able to play a role in that sense. So given that, I moved into, I really want to know which ecosystem or which continent actually serve as the major source of this group, and of course, which one receives more. Of course, we must have this idea before in the previous slide that Asia happens to be the main source, even though it originated in Africa. So most of those things, at some point, they moved into Asia, and then Asia now, since it has connection with America, it gives to America, gives to also some part of the Australia and also the other part of Africa as well. And we'll look into this as well. So North America happens to be the main sink. That means they get more of this species. And it's not really surprising, like I said, from the Beringia land bridge, that's one of the most promising routes. And of course, you see there's a very short distance between South America and Africa as well. So as they come in here, so it's kind of like make it more, the migration much more easier. And so this is another aspect actually talking about what if climate change, what is going to happen? So as individual, definitely these are different climatic parameters. except this ELEV, which is elevation. What this is telling us here is that each of these environmental parameters actually have different role in predicting or influencing the species richness of this group. So when we example is, I'm going to talk about this first two here. Even though amount of annual mean temperature, which this is this AMT here, is very influential, but it's still not significant. Compared to here that, almost all of these are very significant, which means that if there is a significant decline or alteration in all of these climatic variables, we're definitely going to lose. There's a possibility for us to lose some of the species in this group. And so that brings us to the application of this stuff in conservation. So we look deeply, and of course, like I said, so the same interpretation goes for all of them. And why I so much rely on probably, effect of climate to predict this species distribution is because when I correlated the species richness of each of them with longitude and latitude, it really does not, no correlation between them. So this also justifies that the legume, or let me say the MP clade, actually have the opportunity for them to overcome barriers. And that is one of the main reasons why we really have them being distributed all over in the present world. And so this is just like a recap of everything that we have talked about so far, the movement in time and space. So these are the lineages that I talked about that we now defined. And the stem happens to be the origin, that is the age of the group. And then crown means when they began to move. So we can see these are the dates period that coincide with the, with the epoch period that I showed at the beginning. Sorry, these are a lot of jargons. I'm sure many of us will be able to understand this because we've been through this, but these are just predefined. It's just like a calendar. By the time you date your tree, you're just gonna go to geological time scale, and then you just print out this epoch period from there. And this next column talked about the shift. Remember me talking about those dots in the diversification rates, how it's kind of like serve as an indication of heterogeneous diversification patterns. So some lineages do not have, while some really have, and this lineage 6 has 7. And that actually makes it much more, it makes sense to me why it happens to be the largest lineage of them all. And I'm just going to jump to this event route, which is actually at each of the nodes. This is like a movement from Africa, and then dispersed to Africa again, within Africa, and then also vicariance also happen within this particular period. So this is just showing the event route, how this species actually, came into existence as, as a person. I mean, sorry, as an individual, right? So this is how it is. And of course, this is just like the probability that tells us that this movement is actually 100% accurate from the method. And of course, we see like this, for this lineage, this is a little bit more complicated, and it's as maybe like, less than half of like 50% being supported with it. And in summary, I just want to tell us that the global cooling is one of the main factors that actually influence the current distribution of this group. And when we talk about the conservation of this group, the tropics needs to be a center of attention, because both the museum and the crowdful events actually happen within that region. And the assemblage of this species is not far-fetched from it being able to utilize the classic biogeographical patterns, which is mostly, in this case, dispersal events. And lastly, we could see how independent events of speciation and dispersal actually takes place in this particular group. And this is just a model that can also be applied to different organisms if you are really interested to know how they have come, how long they've come in their evolutionary history. So this is just like a take home for us. I'm sure probably we got lost in the process. I'm not sure about that, if I'm making sense at all, but this is just like a compressed summary of everything that we talked about, where I said we have, for the phylogenetic framework, we have now 6 well-resolved lineages within this group. And then we now have like an information about how did it come about. We can see like there is a simultaneous divergence. And of course, the kind of evolutionary event here is actually mixture. There was no homogeneous part, in it. And of course, there is a kind of a linkage between the earth events, historical events, with the evolution of this species. And lastly, this particular group, like I said earlier on, actually serve as you know, a model to test different hypotheses. And as we could see that we could not only use one hypothesis to actually explain the diversification of this group, and we could see that we have almost all of those hypotheses that I actually mentioned at the beginning of the presentation, being one of the reasons why this species has become like cosmopolitan globally. So to that, I would like to thank, a lot of folks that have contributed to my research through time. So this is my, PhD lab in China, and of course, then Arkansas, and here I am right now. And these guys here, they are like my individual guys that, when I need help in, probably analysis or stuff like that, they come through. And of course, I would like to say thank you to everyone for, being here to listen to this talk. And while you ask me questions, definitely you could see some of the, source of fundings for my research. Thank you.
Okay, we have about 10 minutes for questions for the benefit of the recording and those attending online. If you can raise your hand and I will bring the microphone to you if you have a question.
Thank you, Tola. Beautiful and very interesting talk. I, as usual, have many great questions that come from your wonderful talk. I'm just gonna do one so that students can also take the time, maybe to have some questions. So this clade that you work includes the beans that we eat, right? So I was wondering, how did you account for that? In terms of the phylogeny, because there has been a lot of domestication processes, and right. So did you not include those species, those plants, or how did you do, like, how do you account for it being such an amazing crop that has been going around, right?
Yeah, thanks. Thank you. That's a good question. So one of the things that I also noticed is because when I was sampling for this species, on NCBI, you see Phaseolus X. So when I see any of those, even to variety level, I excluded them from the analysis. I just retain only the genus and the defined specific epithets. So anyone that has history of hybridization in them, I just exclude them from the sampling. And if you still remember the last slide, that was the reason why I was like, I really want to know how reticulation actually influenced this topology. And that, of course, will take us through the genomic, maybe like capture data set or something to really be able to deceive us.
Thank you.
Yeah, wonderful presentation, Tola. Congratulations. Can we go back a little bit on the slide where you presented about the map of the world and the biogeographic routes? Yeah, that one with the monkey. This is both amazing and baffling. Um, yeah. So I know this is still in its probably infancy, I would say. Like you still have a lot of areas that you have not gotten samples. So if you increase the sampling, how would that break or test the linkages between these biogeographic regions?
Yeah, thank you for that. From my experience and also, from literatures, I wouldn't expect any breaking from here. I only expect probably more movement between other part of the world and Europe. Because like with 700 plus samples, that's, let me say, still close to probably 40% of the population. I think the inference is quite strong. And my initial hypothesis was that we're not going to have more than 3 lineages in this group. And we see after adding that, we have 6 of it all, right from the deep nodes. So with that, I'm not really expecting too much of change in this, except from the probably between the exchange between other parts of the world and Europe. However, we still need to like do the sampling and see what's going to happen. But that's just going to be my own hypothesis. Yeah.
Focusing on the African continent and its contribution to Asia and Australia, don't you think that it is because of the Gondwana linkage of this? When they broke up 88 million years ago, Africa and Australia, for example, it didn't benefit a lot. But I mean, India. So probably India might have taken some of the lineages and then dispersed it through Asia. And then it's interesting that there's also a good dispersal of species from Asia going to Australia, but there appears to be a blocking mechanism about Australia dispersing back to Asia because it's more, the arrow is thicker going down rather than going up. But the most baffling for me is that South America directly linked to Asia, and then South America directly linked to Australia, or North America directly linked to Australia. Yeah, I'm just curious about how that would, break or be refined if you increase the sampling. And probably this is using what gene allele data again?
Chloroplast.
Chloroplast.
Yeah.
Yeah, I mean, if you have used, angiosperms 353, for example.
I think for either of the molecular data that I used will not really influence this. Rather, it's going to be the biogeography. So in a way, I would actually, when I was presenting, I used the word stochastic. So that means that anything can happen here. I could be here in South America. Probably I'm a sailor or I do whatever it is. And I'm going to spend quite some time in maybe on the sea, right? And then I put some beans in my bag. Yeah. So that's the reason why I put all these guys there as the factors that have been responsible for the stochastic distribution that could actually happen in that sense. Logically, it makes no sense to see that how would the legume actually be, survive for a longer period of time before it gets sealed to this. Another thing I want us to also think about is time. These things don't just happen at the blink of an eye, right? So remember, probably at some point it gets stuck at a particular point, it continues its life there, and then after some time, it also find another dispersal means that move to another certain distance, and then it's moved that way. So those are some of the things that actually make legume diversification very complicated. Unlike some species that, for instance, I don't know if you know, some species like ficus, that they are actually very big. They are not, they are bigger, the seeds are bigger, and also they don't even stay longer viable, and they can also stick. Some of the groups in this particular clade can also stick to animals, furs or feathers. So they actually make use of multiple dispersal routes to acquire or to get to where they are right now. And that has been supported by the elevated dispersal rates in the group.
So question for you. Thank you, Tola. That was great. The graphics were incredible for describing. You did a great job of explaining them to those of us that don't work regularly with those. I still have a lot to learn. But with that said, speaking from the herbarium perspective, if this herbarium were to have an excellent collection to support your work, what would that look like in terms of the age of specimens, diversity of all that good stuff?
Yeah, with reference to this group, actually, I still feel more like I need to get more samples from, still from Europe, because I spent about maybe 4 to 5 months sampling NCB high. That's one of the gene banks that we could get molecular data from, and also other archives. I actually have very less representative from Europe. And although there are a lot of argument that has also been going on that, of course, legumes cannot survive those harsh cold, and then that's why they moved down around that period of time. But there are some fossil evidence that legumes have actually existed even before the 60 million years ago, around that Cretaceous period, right? So I would probably focus more on, species distributed in Europe. And of course, if I can't find them even in gene bank, it means that's a lot of things that probably might actually give everything a new face.
Thank you.
You're welcome.
I've built on those exchange partners.
If no one else has questions, I have two quick to follow up on your question, Tiana. I was curious just generally where the center of diversity for the MP clade is worldwide. Is it for example, is it in Asia? And might that explain why we see this bias toward much greater dispersal from Asia? And it just happens that right now, this is where most legumes, beans are currently known from, where the highest species richness is globally?
I think from the very maybe in the middle of my presentation, just a little bit more. So this is actually modeled based on the current distribution, and this is clearly like this doesn't have molecular data, right? So we look at them from different perspectives, and I expect that this is more richer because I only visited GBIF, downloaded the occurrence coordinates, filter it out, and then have this. So technically, this is telling us more about the distribution pattern, hotspot regions, which I also think has a little bit interpretation to the molecular data that I have here. So basically, I would just say there is no one hub or hotspot for this group. We have them being scattered all over the places, but particularly we could see basically in the probably southern hemisphere and around the equator axis. So it didn't just go beyond that.
Cool. Awesome. Thank you. And then I mentioned I had a second question. The other one, I'm glad you took it back to the slide. That was part of my plan, is when you have the clades that have a bipolar distribution that are like amphi-temperate, one in the temperate north and one in the temperate south, you talked a lot about diversification rate and adaptation, or the ability to adapt. I'm curious about these clades in particular. Are there any traits or characteristics about them that make them better adapted to these temperate environments that you could give an example of? And as a result, are they sort of stuck there, or if they just specialized there and they outcompete other temperate lineages or other lineages that are better adapted to the tropics instead of temperate habitats?
Thank you. That's a good one. So I'm going to answer the question from probably from this slide. So the first thing we need to think about is this whole genome duplication. And this actually has been linked with genes responsible for the evolution of nitrogen fixation in legume. So outrightly, even if they find themselves in a very harsh condition where there are very low nutrient, definitely this is going to come into play. And I would also hypothesize that the number of whole genome events that was detected actually may have a strong link with probably their distribution, or let me say the ecological conditions at that point in time. So I may not be able to tear apart which lineage actually have, more of these nitrogen fixation genes in them, but it's actually common in the ancestry, which is very key in their evolution. Once we have it here, then it began to, occur again and again, and also make those things, make them more adaptable to different climatic conditions. And I would say that the species that actually have bimodal distribution may actually have the opportunity to actually do that. And also that particular group, their seeds can actually attach to, to animal skins. So while some could actually survive in a temperate, some can move down, and also, like having those two distinct lineages. But surprisingly, I could not actually, look at the maybe biome classification of it, because with that, if I follow biome, probably I will be able to see the clades that are actually concentrated in the tropics and the ones that are in the temperates. So that would have, given us more clue into your question. And of course, probably if I still have more time, I could it's just about mining data for the biomes, and I just do the estimation. Yeah, that's a good insight. Thank you.
Yeah, great talk.
Yeah.
Alrighty, with that, we are actually just a little bit after 1 o'clock. So I think Dr. Oyetola can stay for a little bit after if you have any more questions, but thank you guys so much for attending.